when patients don't have driver mutations for non-small cell lung cancer, the current frontline approach is immunotherapy given by itself or in combination with chemotherapy. Multiple studies have showed that chemotherapy given with pembrolizumab or chemotherapy given with bevacizumab and atezolizumab are effective strategies in the frontline setting. The other strategy that's been under evaluation is the combination of ipilimumab and nivolumab, which is a non-chemotherapy combination, both agents targeting the immune pathway. We know that the combination of IP and Nivo is successful in improving patient outcomes for metastatic melanoma. In non-small cell lung cancer, we have seen data from phase 2 studies earlier that showed promising outcomes in terms of response rate and progression-free survival. The CheckMate-227 trial was specifically designed to ask a few questions. It's a large trial with close to 2,000 patients enrolled. One question was to compare the outcomes of EP plus Nivo to either Nivo alone or chemotherapy in patients with high PD-L1 expression. That group enrolled about 1,150 patients. The next group was for patients with low PD-L1 expression, where the randomization was to Nivo plus EP versus chemo alone versus chemo plus Nivo. So one of the arms in the PD-L1 high and PD-L1 low were slightly different. What we are looking forward to is seeing the results of the outcome with EP and Nivo based on PD-L1 expression. We had seen data based on tumor mutation burden last year at the AACR meeting and subsequently in a New England Journal publication, where if patients had a higher tumor mutation burden defined as 10 mutations per megabase, the combination of EP plus Nivo was superior to chemotherapy alone in terms of PFS. But what we later found out is that those results were also seen in patients with low TMB, which raised questions about whether TMB itself is a good predictive marker or is it just a prognostic marker. The CheckMate-227 trial that we're looking forward to hearing about in the next few weeks is going to report on the PD-L1 expression as a predictor of outcome with EP plus Nivo. The press release from the sponsor noted that the study met its primary endpoint, showing superiority for EP plus Nivo in the high PD-L1 group compared to chemotherapy alone. So at the meeting, we obviously would want to know what is the PFS improvement with the EP plus Nivo combination, what is the impact on overall survival, and also more information on the safety profile. This being a very large trial, we will have enough information to do even subset analysis to understand other questions on top of the top-line biomarker question. My hope is that after the results are reported, we'll have more clarity on which group of patients can get IO alone, which subset of patients need chemo plus IO, and is there a subset of patients that can get just IO plus IO combination for frontline therapy of non-small cell lung cancer. When the EP plus Nivo regimen was initially developed, the main question was, will it be safe enough for patients with lung cancer? And as a response to that question, the investigators looked at various dosing schedules and came up with a dosing of Nivo given at 3 milligram per kilogram every 3 weeks and EP given at 1 milligram per kg every 6 weeks. And with that regimen, the tolerability seems to be very good. This is very different from the dosing schedule that's used in the melanoma setting, where more EP is given compared to relative Nivo dose. Now that we know that this regimen is relatively safe, looking at the efficacy outcomes based on PD-L1 expression would, I believe, shed new light on optimal role for this agent in the setting of non-small cell lung cancer. And eventually, having more options will allow us to individualize therapy to patients based on biomarker status, patient preference, toxicity profile, and ultimately the cost of the care as well.